Welcome to the problem-solving set on conjugation. Question 1. The results from an interrupted mating experiment shows that gene DM first appears at 15 minutes, gene GB first appears at 3 minutes, and gene NIC first appears at 7 minutes. What is the order of transfer? So when you're talking about conjugation, you can figure out the order of transfer from donor cell to recipient cell by seeing which gene appears at the earliest time all the way up to which gene appears at the latest time. So the order of transfer, the first one that appeared was gene GB, because you see that 3 minutes is the earliest time a new gene appeared. So the first gene is GB. Then the next earliest time you see is for gene NIC, which was 7 minutes. And then finally the last gene to transfer was gene DM at 15 minutes. So this is relatively simple. Again, remember, I'll write the order of transfer here, going left to right, and that's from least amount of time that it appears to the latest time, or most time that it appears. Problem 2. In a cross between HFR and F-, the PYRF gene enters the recipient cell in 4 minutes, a point in time where there are no X conjugates that are TRP plus, GLU plus, VAL plus, LU plus, or ASN plus. The mating is then allowed to proceed for 20 minutes, and PYR plus X conjugates are selected. Of these cells, 34% are TRP plus, 5% are VAL plus, 62% are LU plus, 96% are GLU plus, and 1% are ASN plus. What would you predict is the order of the genes? So first let me just define quickly for you what an X conjugate is. I'm not sure if that was covered in the theory videos, but an X conjugate is the resulting cells after conjugation has taken place. Okay, so this problem is saying that when you're selecting for PYR+, that is an early marker that is transferred from the donor into the recipient cell through conjugation, which is through contact between the cells, at an early time. So that happens at 4 minutes. So the first gene in the order that we're being asked to predict is going to be the PYRF gene, right? Because that's the one that is being transferred over first. So the order here is going to be PYRF. Now, in order to determine the rest of the order, what you need to know is that the contact between cells during conjugation is very fragile. So the farther the gene is from the PYRF gene, the more likely it is that that fragile connection between the two cells will be broken before that gene can be transferred. So therefore, the frequency, which is the percentages that they're giving us, the frequency of these genes beyond PYRF are transferred decreases with the distance from early marker. So the farther away it is, the frequency is going to be lower. So let's look at these frequencies. What is the highest that you see? You see, get the highlighter, 96% seems to be the highest frequency, and that's GLU plus. So we're going to say that GLU is the next gene in order. The second highest frequency, if you scan through, is going to be the 62% for leucine. So the next gene is leucine. Then after that, it would be 34% for tryptophan. So the next gene would be TRP. Then finally, you have the 5% for valine and the 1% for ASN. Okay, so this is the order of the genes. So this is the earliest to be transferred and the latest to be transferred. And remember, the reason is because contact is fragile. So I'll say as distance from marker, as this distance increases, the frequency will decrease. Okay? So just to reiterate, I want to make sure that this is clear. The reason, let's say, you only have 5% of these X conjugates are Val plus. That's a very small percentage, right? So they're saying that they're only allowing this mating to happen for 20 minutes. PYR plus gene transfers over at 4 minutes. So after that, you only have 16 minutes for a transfer to happen. Now it's saying that because the contact between cells are fragile, and as we predicted, valine and the PYRF gene are very far apart, there's 3 genes in between them. Because they're so far apart, there's much more likely chance that the contact between the cells will be broken before the valine gene is transferred over. So that's why there's a few percent happen, because not all the connections break at the same time. So some of them are strong enough to keep the cells in contact for that gene to be carried over, but it's not going to happen all of the time because they are so far away from each other. Okay, so as distance from the early marker increases, the frequency for X conjugates with that gene will decrease. Question 3. An F- strain of E. coli is resistant to ampicillin but auxotrophic for alanine, cysteine, glutamine, proline, and valine. A series of interrupted mating experiments are run with 2 different high-frequency recombination strains, HFR1 and HFR2. Both HFR strains are wild-type for all amino acid markers, but are ampicillin sensitive. The data below describes the time in minutes at which recombinants containing the indicated amino acid marker were first observed. Sketch a chromosome map that includes all of the markers, the distances between each marker, the position of origin for each HFR, and the direction of transfer of each HFR. So let's look at each strain of high frequency one at a time. So for HFR1, we want to look at the order of transfer. So the order of transfer of the 5 genes is indicated by the time at which cells showing the marker first appear. So the first one that you could see that showed up at the earliest time was the alanine marker at 5 minutes. So I'll write O for origin of replication, and then you have 5 minutes until you hit alanine. Then the next would be valine, and between 10 minutes and 5 minutes, that's a 5-minute gap. Then the next up would be proline. Between 10 minutes and 35 minutes is a 25-minute gap. Next would be cysteine, and that is a 30-minute gap. And then finally the last marker is glutamine with another 10-minute gap. Okay, so although the chromosome map that we're going to draw is going to be circular, I just want to write this linearly to show the order of transfer of each of the markers. So now for HFR2, we have the origin, and the first marker showing up is proline after a 10-minute gap. Next you have valine after 25 minutes. Then you have alanine at 5 minutes. Then glutamine, let me see, so 20-minute gap. And finally cysteine with a 10-minute gap. So as you can see, the HFR1 goes from alanine, valine, proline, cysteine, glutamine. But if you look, say, at alanine on HFR2, you can see that in order to go in the same order, you would have to go the opposite direction, right? You'd have to go to the left to get valine, proline, and then loop around to get cysteine and glutamine. So it's obvious that the origin of replications for both of these strains are going to have different directional arrowheads to them, right? So let's draw a circular map. That is going to be the F factor plasmid. And let's draw HFR1 first. So I'll draw its origin of replication in red. And you draw it as a triangle that kind of looks like a play button. Now just so you know, in case you see these maps on an exam, or you have to draw one on an exam, that when the origin of replication is drawn this way, it means that you're going to be transferring the genes in the counterclockwise direction. So you're basically you're going in the direction that's opposite of the point of that triangle. Okay? So if it was drawn this way, that means the order of transfer would be going that way. Okay? Just so you know for future reference. So the first gene marker that we have for HFR1 was alanine. Then you had valine, and this had a 5-minute gap. So I'll draw like this and draw 5. Then you had 25-minute gap to proline. Then you had a 30-minute gap to cysteine. Draw it a little bigger. And then finally a 10-minute gap to glutamine. Okay, oh, so let me label this in red. This was HFR1. Okay, now for HFR2, the first marker that appears for that strain is the proline. Now there's a 10-minute gap in between, and you're going to be going clockwise for this, right? Because you have to go proline, up to valine, alanine, etc. So let me draw that one in blue. So we're going to draw the origin of replication before proline, and we're going, remember, we want to go in the direction that's opposite the point of that triangle. So we're going to draw it like this again. Seems a little counterintuitive, but remember that this is a circle, so on the bottom, this means that we are going to be going this way. Okay, so you have a 10-minute gap to proline, then 25 to valine, that makes sense, 5 to alanine, 20 to glutamine, and then 10 to cysteine. So this is HFR2, and it fits well with our chromosome map. So this is the complete chromosome map that includes all 5 markers for both HFR strains. Okay, so we showed, we include all the markers, we included the distances, the position of each HFR origin, and the direction. Okay? Now part B asks, if another mating experiment was done using the same strains and interrupted after 60 minutes of mating, what type of medium can be used to select for progeny recombinant for the proline gene, Pro+? So in order to do this, remember that you want to just select for the recombinant offspring. The original HFR strain are wild type for all amino acid markers, but those are parental, right? They're not recombinant. So what we want to do is select against the HFR strains. So in order to do that, we would need to plate on ampicillin. So that would select against HFR because the HFR strains are sensitive or not resistant. So I can put that there, AMP superscript S, and that's for sensitive. And we want to only see the progeny that are wild type for proline, and that can make proline themselves or prototrophic for proline. So in order to do that, you want to leave proline out of the selective media. So in order to do that, we want to plate everything else in that media. So we want alanine, the other 4, cysteine, glutamine, and valine. Okay, so because only proline is left out, that means that you're selecting for the recombinant progeny cells that have received that wild type marker from the donor cell, the HFR strain. Okay, so all of this put in the media to screen for the Pro+ progeny. Question 4 is just an easy application of the theory. So it says, out of the following choices, choose which best describes a bacterial cell with 1. Phenotype F+, 2. Phenotype HFR high frequency recombination, and 3. Phenotype F-. So for part A, the description is that it can donate an F factor during conjugation. So this describes a bacterial cell with the phenotype, and really the genotype, of F+. B says that it can receive the F factor during conjugation, and that is for a bacterial cell with the phenotype and genotype of F-, right? They're the recipients of the donor F+. C says can donate some host chromosomes during conjugation. Now this is for the high frequency strain, right? That's what it describes. so HFR, and D can receive some host chromosomes during conjugation. And again, this is the F minus. So this is just application of theory. F minus are generally the recipients, F plus are the donor cells, and HFR are also donor cells, but they can bring along and donate some host chromosomes during the contact of conjugation.